fermented meats and the implication in human health. And I'm a microbiologist, and so I'm interested in this microbiome connection. So just an overview of the talk. I'll start out with talking about population human health trends that have been associated with meat consumption, and then go on to talk about what exactly fermented meat is and how the microbiome, or all of the microorganisms that are found in fermented meats, play a role in meat fermentation, and then go on to talk about the ingestion of fermented meats and different microbial mechanisms that may be related to some of the variations that we see in health trends. And I'll be focusing mostly on microbial sources of precursors of nitric oxide, and N-nitroso compounds that have been associated with increased risks of cancer. This is from a paper by Daniel et al. That shows in the last 40 years or so, actually meat consumption has increased. And you can see here that in the US, we are leading the charge on meat consumption. And it turns out that the majority of this meat is red meat that's being consumed in greater quantities than poultry and fish. And there's been some work done in the NHANES cohort which suggests that up to 22% of this meat consumption may be due to fermented or processed meats. In 2007, the WCRF and AICR came out with a review of 16 cohort studies and 71 case controls, which suggested that there was a positive association between the consumption of red and processed meats and risk for colorectal cancer. They also said that there was a probable association between fermented fish and their nasopharyngeal cancers. And their recommendation was to reduce red meat intake to about 500 grams a week and not to consume any processed meats. This caused a large uproar in many ways, and one of the biggest criticisms of this report was that there were some methodological problems, and that there needed to be some more accurate tools for assessing and quantifying processed meat intake. So in 2007, Cross et al. Did just that, and they looked prospectively in the NIH AARP Diet and Health Study, which has about 500,000 subjects. And they increased their tools for assessing dietary meat intake and risk assessment by using a food frequency questionnaire, which most people have used. They also used a risk factor questionnaire, which looks at lifestyle and health risk factors. And then they also used updated databases for dietary nitrate and nitrite consumption, and also a new database which quantifies products associated with charred meats. And when they looked at the lowest consumption quintile and the highest consumption quintile, they found significant associations between the intake of heterocyclic amines, precursors for N-nitroso compounds, which are nitrate and nitrite, and also intakes of iron, either as dietary iron or heme iron. So what is fermented meat? This falls into the category of processed meat. And processed meat is loosely defined as meat that has been preserved by smoking, curing, salting, or adding chemicals like nitrate and nitrite. And cured meats include entire pieces of raw or cooked muscle meat, and they use either nitrate or nitrite as a dry salt solution or a salt solution. And then there's a process in which ripening occurs and drying occurs, and it changes the nature of the meat. And one example of this is prosciutto. And then the other area that we're focused on are raw fermented sausages. And in this case, uncooked coarse mixtures of lean meats and some fats are ground together and put in casings. They're salted, or nitrate or nitrite might be added. And this is also characterized by having a ripening process or a drying process associated with it. And these are characterized by having a large period of time in which microbial anaerobic fermentation occurs. And sometimes you can use starter cultures which change the nature of the bacteria associated with the fermentation. And one example of this is salami. So why have we fermented meat? It's been around for a long time. And fermented meat is fermentation that occurs by anaerobic microbial metabolism by either bacteria or yeast, which we just found out about in the last talk. And one of the reasons that we ferment meat is that it boosts acidity, so the pH increases in the fermented meats over time, and this tends to reduce pathogens. It can also counteract rancidity of the fats that are in the fermented meats. And microbial end products also help to enhance the flavor, the taste of the meat. There's some texture improvement that occurs through the proteolysis of the proteins in the meat and amino acid fermentation. And this can occur with or without salts and nitrate or nitrite and smoking. Over time, there's been a movement towards using nitrite as the curing agent of choice. And one of the reasons for this is that it's been found to affect the quality in a more standardized way. So over the fermentation over the fermentation period, there's a pink coloring that develops, and this is specifically related to the formation of nitric oxide and myoglobin. And it can also be used, the formation of this pink color can be used as a visual indication that the fermentation process is occurring well. It also affects the flavor. So nitrite can control lipid peroxidation, so it guards against rancidity. And then also it can select for different kinds of microbial fermentation, and those fermentation end products affect the taste of the meat. There are also microbiological safety issues that are associated with this, and there are really antibacterial effects here. And that changes in the pH, high salt, and iron content can select for different organisms and select specifically against pathogens. So this might be species specific. For example, there's a reduction in gram-negative enterics like E. coli and Salmonella. And also there's a change, or there's a protection against Clostridium botulinum, as nitrite specifically inhibits the cell division in vegetative cells. And then also in combination with a prebiotic like lactate or acetate, you can have protection against Listeria pathogen, the pathogen Listeria monocytogenes. But of course, there have been health concerns associated with using nitrite, and this has been a large topic of debate. And the US and the EU and other countries have limits on how much nitrite is allowed to be used. And this is because nitrite is reduced to nitric oxide, and that can combine with secondary and tertiary amines, which are formed during protein degradation, to form N-nitroso compounds. These compounds then form alkalizing agents which attach to DNA and become genotoxic if there is no causing mismatch repair, that kind of thing. So there are many different types of fermentation that are carried out by microbes, both eukaryotic microbes like yeast, the Saccharomyces, or by bacteria. And so Sharon very nicely covered the fermentation by yeast of glucose to ethanol. There's also acetic acid fermentation by Acetobacter, in which ethanol is formed, acetate is formed from ethanol. There's also lactic acid fermentation, in which glucose is converted to lactate. or lactose is converted to lactate. And this is carried out by a phylogenetically broad group of bacteria, like Leuconostoc, Lactobacillus, or Streptococcus. And then there's also heterolactic fermentation, in which glucose is decomposed to either ethanol or lactate. And then there's also alkaline fermentation, in which proteins are broken down to amino acids and ammonium. We'll mostly today be concerned about the lactic acid fermentation and the alkaline fermentation, which are two fermentations which occur in fermented meats. The other aspect that I think is not often talked about is the nitrogen cycle in fermented meats and within the human body. And this is really carried out by bacteria, by eukaryotes or human cells, and also there's an abiotic aspect of this metabolism. Bacterial nitrogen metabolism in this case is associated with dissimilatory nitrate and nitrite reduction. And in this case, very specific bacterial enzymes by anaerobic bacteria convert nitrate to nitrite with a nitrate reductase, or they convert nitrite to nitric oxide by a nitrite reductase. And then there's also a modification that can occur. In humans, there is a very different system that produces nitric oxide, and it's using aerobic metabolism as opposed to anaerobic metabolism without oxygen. And in this case, arginine and oxygen decompose to nitric oxide by using nitric oxide synthase. And then there's also a very important process in the body, which is just abiotic formation of nitric oxide. In this case, nitrite is reduced to nitric oxide through disproportionation at pHs of less than 5. So these are the nitrogen pathways that are important in fermentation. So when we look at microbial metabolism that may be associated with the dietary intake of fermented meat, it happens at very different, in many areas of the human body and also within the fermented meat itself. So here's an example of a person, cross-sectional analysis, who's dreaming of all of the different fermented meats he gets to eat. And there's fermentation which occurs within the meat itself, within the mouth by bacteria. Then there's abiotic disproportionation of nitrogen compounds in the stomach. And then within the gut, there's also formation of nitric oxide and N-nitroso compounds by bacteria. So first we'll look at the microbial community associated with fermented meats. And this is a study by Polka et al., and he looked at 6 different vendors of salami makers and followed the changes in the microbial community over the period of time in which they mixed the meat and put it in casing, and then followed the microbial community through the drying period. This was the microbiome, or all the bacteria that are in the microbial community in the meat were measured using 16S ribosomal RNA, the gene from that. And that really gives us an idea of the full complement of the bacteria that are present and the diversity of the bacteria that are present, as opposed to doing pure culture approaches. And this is a cluster analysis of the results. And then the contribution of each of these different bacterial families are listed here in the bar graph from 0 to 100%. And the colors at the bottom are the different families that were measured using this molecular biology tool by sequencing the 16S ribosomal RNA genes. And what they found was that at time 0 here, all of the samples clustered together, which was interesting. So their community, the microbial community that starts out at the beginning of the mixing period is very similar. You can also see that it's very diverse and that there are many more different colors that are in this cluster. So it's dominated by, there's Brevibacter, Lactobacillus, and Staphylococcus, and then many other organisms that are there. And then over time, there is a shift towards lactic acid fermenters. So this is both present in the Staphylococcus up here and in the Lactobacillus, which is shown here. And this happens as there are changes in the physicochemical properties of the sausage, which really imparts the flavor in what we all enjoy, how we all enjoy eating the sausage. So over time, the microbial fermentation and the drying produces changes in the water holding capacity of the sausage. And then also the pH is reduced, so it becomes acidic over time. During this period, actually, there is disproportionation of nitrite if the sausage is cured with nitrite, and nitric oxide can be formed because of the acidity in the pH, in the lower pH. Then moving on, there's also a large component of nitrate reduction which occurs in the mouth, which is really a synergistic interaction between the human host and the microbiome. And this is a larger cycle which is integrated across the human host, which is called the enterosalivary nitrite pathway. That's a mouthful. But there was some interesting work by Peterson et al. In 2009 who showed that if you give somebody some antibacterial mouthwash, that there was a reduction in the nitrate in plasma, and this was associated with increased blood pressure. And so this is part of a cycle which starts with the ingestion of dietary nitrate. In the small intestine, this nitrate can enter into the bloodstream and circulate, where it also combines with supplies of endogenously generated nitrate, which were generated through the nitrite synthase pathway. And then up to 25% of the circulating nitrate in plasma is actively taken up by the salivary gland and concentrated. This is then dumped into the mouth. And it turns out that the mouth have a large number of nitrate-reducing bacteria in the oral cavity. And this biological pathway can produce the precursors for NOC compounds. So in this study by Hyde et al., they looked at the tongue microbiome. And when I say microbiome, what I mean are all of the different microbes that live and grow on your tongue. Then they used 16S ribosomal RNA gene sequencing to identify all of the bacteria that are here. And they looked in high nitrite producing people and low nitrate producing people. And this is just a bar graph of the number of different kinds of organisms that are present, and the percent abundance is on the x-axis. And what they've discovered is that there's a microbial pathway that's independent. It's nitrite oxide synthase independent and oxygen independent, and forms the precursors for NOC. And this is carried out by the oral bacteria, where they reduce nitrate to nitrite using nitrate reductases. What they found when they looked at the tongue microbiome, that in the high producers, there was a dominance of Streptococcus, and then similarly, the low producers also had a dominance of Streptococcus. However, the rest of the microbial community was very different, and you see that there's a much more diverse microbial community associated with high nitrite producers, as opposed to the low nitrite producers, which were dominated predominantly by Lactobacillus. They also then went on to look at the microbial genes that are involved in the actual process of the reduction of nitrate to nitrite, and looked for the genes, gene clusters of nitrate reductases and found that indeed there were more genes associated with nitrate reductase in the high nitrite producing microbiome, which so then this is another source of nitrite, which is then generated in the mouth and then swallowed into the stomach. So in the stomach, the nitrogen cycle is dominated by disproportionation and NOC formation. And this pathway has been around a while. So what happens is that the nitrite that was formed in the mouth enters the stomach, which is around pH 5. And at this pH, nitrite disproportionates to nitric oxide. And there's also dietary protein metabolism occurring here, which leads to the formation of amines and amides. And then these combine to form N-nitroso compounds, which is shown across the bottom here. These N-nitroso compounds then enter the gut, and they're hydroxylated by the human enzyme CYP450, and they become alkylating agents then, and they form DNA adducts, which can cause mismatch and miscoding if not repaired, and lead to carcinogenesis or tumor formation. And finally, we get to what happens when in the gut when you eat fermented meats or high protein diets. There can be nitric oxide formation or N-nitroso compound formation. And the thing that's interesting about this is that our gut microbial community is very diverse. If we were to take a fingerprint of the gut microbial community of every person in this room, you would have a different signature. And that can affect how your body, how the microbiome metabolizes your diet, and then what chemicals you are exposed to. So we each have a different individual exposure potentially. And in microbial nitrosation in the gut, there's about 30% of the dietary intake makes it into the gut. There's protein digestion to amines and amides. And then in a very early study, people at first didn't know if intestinal bacteria would be involved in nitrosation. But in an early study by Massey et al., they looked at the apparent total N-nitroso compounds in conventionalized mice, or mice that have a fully diverse microflora, or microbiota, and germ-free mice, which are mice that don't have any bacteria in their gut, and they fed them nitrate, and they found that there was an increase in the ATNC in the mice that were conventionalized. There's also been some further studies that have looked at the activity of nitrate reductase, or those specific bacterial enzymes associated with converting nitrate to nitrite in the gut, and they find that they increase along the GI tract in conventionalized rats. And then also, there are specific bacteria which are capable of actually creating these N-nitroso compounds, such as gamma proteobacteria and pepticoccus. So this is a study from Ruth Lay et al. And really, what she did was, the take-home message here is we are what we eat. She looked at different mammals with different dietary habits. So she looked at herbivores, omnivores, and carnivores, and then measured the gut microbiome from each of these different species of animals, and found that they cluster by their dietary habit. And here you can see the carnivores are down here, and they're clustered together, suggesting that there's selection of a microbiome which is associated with and tailored to the decomposition of meat products. And the gut microbiome and the composition of the gut microbiome can affect exposure of the human host. And in this case, we're looking at the effects of meat ingestion on the production of N-nitroso compounds. And then this is a dietary intervention done by Russell et al. In 2011, and they recruited 17 obese men, and they used a randomized crossover design, so they fed 3 different diets over time with an appropriate washout period, which is a great experimental design in human studies. And they fed them either a maintenance diet or a high protein diet with medium carbohydrate or a high protein diet with low carbohydrate. And here you can see the medium diet had 13% protein and 50% carbohydrate. The high protein medium carbohydrate diet had 28% protein and 35% carbohydrate, whereas the high protein low carbohydrate diet had a 29% increase, or 29% protein intake and a 5% dietary carbohydrate. And what they found across the 3 different diets is that the N-nitroso compounds increased as a function of protein intake and shifts in from medium carbohydrate to low carbohydrate. And on this y-axis here are the total N-nitroso compounds that were formed. So this diet, which was high protein and low carbohydrate, formed the most N-nitroso compounds. And really this was associated with a shift in the microbiome and with metabolites in the diet. And really the take-home message here is dietary composition matters. So you may be exposed to different environmental compounds which are carcinogenic, but really it matters within the milieu of what your exposure is as to the effect. So in this case, this is a multivariate analysis of both metabolites and the composition of the gut microbiome. And you can see that this high protein, low carbohydrate diet, the microbiome and its metabolites cluster separately from the other diets. And what this dietary intake was characterized by having much higher N-nitroso compounds. There's also a reduction in butyrate, which is a short-chain fatty acid fermentation end product, which is associated with positive health outcomes. And there is also a decrease in the bacteria that are associated with Rosaburia and Eubacterium rectali, who produce the butyrate. So for this talk, I've focused mostly on precursors for N-nitroso compounds, which then can lead to tumorigenesis. But there are other aspects of dietary intake associated with fermented meats that can also be genotoxic. And this is a recent study from Martin et al. In which they looked at heme iron, lipoperoxidation in the gut microbiome. This pathway is associated with the heme iron induces lipoperoxidation, and this forms malondialdehyde, or MDA. MDA is also another compound which can form DNA adducts and then lead to tumorigenesis if those adducts are not repaired properly. And in this experiment, they were interested in looking at different types of heme or iron compounds, either as hemoglobin or hemin. Hemoglobin is associated with ingestion of fresh red meat, and hemin is associated with the ingestion of fermented meats. And they fed the rats antibiotics, and what they found was that the antibiotics reduced lipoperoxidation. And on this side of the slide, you can see here's the treatment without antibiotics. Here's the treatment with antibiotics. And on the y-axis are the MDA forming, the number of MDA equivalents formed. And what you see is that when these mice ingest hemoglobin, there's a large number of MDA equivalents formed and a significant increase in MDA equivalents when hemin is ingested as well. And then when you give the rats antibiotics, those effects, or those compounds that can potentially form DNA adducts and are genotoxic, are reduced. And so this suggests that there's an interaction between the gut microbiome and the formation of these compounds. And we all know that taking antibiotics alters your microbiome. And it could be that there are changes in the microbial community structure in that, that alter the production of hydrogen peroxide and therefore alter lipoperoxidation, which occurs. And for example, Bifidobacter, Lactobacillus, and Streptococcus, which are bacteria that are often found in probiotics, are catalase positive bacteria, which means that they produce an enzyme which destroys hydrogen peroxide, and that's associated with reduced lipoperoxidation. In contrast, Enterobacteria acei are catalase negative, and they produce hydrogen peroxide, which enhances lipid peroxidation. So in summary, microbial metabolism in the human system may be another variable that should be used to account for variation in the response, or variation in some of the epidemiological trends that we see associated with fermented meats. And although fermented meats have nutritional benefits with increased iron, protein, zinc, and B12, these consumption of large amounts, especially in a diet that doesn't have other balance, or has lower carbohydrate, may shift the microbiome towards organisms which produce more harmful compounds and may affect human hosts negatively. And this is a complicated system to measure or to understand, because really there's interaction between the human host and the microbiome. And so really we need some integration of biomarkers of meat intake, looking at human gene expression in response to meat intake, then also looking at individual genetic variation in the enzymes that are involved in some of the CYP450 kinds of reactions, and then also looking at variation in the fermented meat metabolism by the gut microbiome. And these require integrated approaches, experimental approaches really, across both human cell lines and gut microbial in vitro systems, and then some in silico studies, and I think that also the use of dietary interventions in these systems where you actually measure both eukaryotic biomarkers of disease and the microbiome are important. Thank you, and I'll take any questions.